For decades, neuroscience operated on a straightforward assumption. We believed the brains of all mammals were built from the exact same biological hardware, a mix of neurons that transmit signals and glial cells that support them. This foundation was established by the early pioneers of brain mapping, who painstakingly categorized these basic cellular building blocks. Because those building blocks appeared identical across species, we assumed the rules were identical too. The working theory suggested a human brain was essentially a mouse brain, expanded in size but utilizing the exact same cellular Lego bricks. But that theory created a tension in the data. If our microscopic hardware is identical to a rodent's, it fails to explain why human cognition, self-awareness, and complex thought are so distinctly different. This assumption has hindered medical progress for years. Researchers have repeatedly tried to solve complex human neurological disorders using animal models, only to see trial after trial fail to translate to the human mind. Our anatomical records were incomplete. We were operating with a map that omitted a critical component, largely because our standard imaging tools were unable to detect it. That began to change with the Allen Brain Atlas Project, a systematic attempt to catalog human brain cells, to identify why human brains don't always behave like their animal counterparts. To do it, researchers had to look past bulk tissue and shift to a technique called single-nucleus RNA sequencing. In the laboratory, this means pulling apart donated brain tissue to isolate and analyze the genetic core of individual cells. Scientists read the unique genetic barcodes of hundreds of thousands of cell nuclei. This allowed them to cluster cells into families based on their specific genetic signatures, rather than just their general appearance. In 2018, the data revealed an anomaly. Researchers found a group of cells with a specific combination of ion channels and synaptic proteins that had never been seen in a rodent or any other mammal. The discovery provided the first molecular proof that the human brain contains cellular hardware that is entirely unique to our species. Under a microscope, these cells look distinctly different from the standard elongated neurons we're used to seeing. They are small and compact, with dense, bushy dendrites that look like the fruit of a rose plant. These Rosehip neurons are located primarily in layer 1 of the cortex, the brain's outermost layer, where information from across the entire brain is integrated and processed. Their mechanical function is even more specific. They possess extremely strong inhibitory power, acting as a specialized brake within the brain's electrical networks. When they fire, they silence large groups of surrounding excitatory neurons with remarkable precision, controlling signal flow through the cortex. This precise timing is the biological basis for our higher cognitive functions. Because Rosehip neurons dictate the narrow windows of synchronized activity in the brain's integration zone, they may provide the cellular architecture necessary for the selective, highly integrated processing that defines human conscious awareness. Finding a regulator that exists in humans but not in mice finally explains why neurological drugs that fix brain circuits in animal models so often fail to work in human patients. This comparison shows a healthy brain on the left and a probable Alzheimer's case on the right. In diseases like Alzheimer's, schizophrenia, and epilepsy, the early loss of inhibitory neurons disrupts the electrical balance, leading to the breakdown of memory and cognition. Since these circuits are human-specific, researchers are now bypassing animal models to study them. They are using advanced human organoids, three-dimensional tissue models grown in the lab, to observe how these neurons function in a living system. The ultimate goal over the next decade is to use tools like CRISPR to therapeutically modulate these cells. By targeting the Rosehip neuron, we may be able to restore the inhibitory balance required to treat chronic neurological conditions. The existence of the Rosehip neuron proves that our internal map still has significant gaps. Every time we correct it, we get closer to understanding the biological substrate that allows us to experience the world.